my name is Tegan Mulford, and today I'm going to be talking to you about the biogeography of this wonderful fly, Proctacanthus. I'm a master's student at BYU, and I'm really excited to talk about this with you today. So this is Proctacanthus. Proctacanthus is a type of robber fly or assassin fly within the genus Proctacanthus, in the subfamily Asilini, as described by Cohen et al. In 2021. And these flies are voracious hunters. They make a marked impact in grasshopper populations and are just generally pretty astounding to look at. Along with that, they're also pretty big. So here's my face for comparison. Um, but they are just extremely large flies, and because of that, they are often collected and remain sitting in natural history collections without proper ID and with just a lot of locality information associated with them. So there's no key to the genus. It's in need of taxonomic revision. That's the focus of my master's. But for this project, knowing we had so much data associated with these flies, and knowing what we can do with ecological niche modeling, I decided to investigate that route. And I had two main questions. The first one is, how well does ecological niche modeling predict the ranges of these flies? And then the second one is, can we use ecological niche modeling to aid in delineating species within the genus? So before we could answer these questions, we have to define what exactly we're talking about and where exactly we're talking about it. Within the genus Proctacanthus, there are two main groups, those with straight mouth parts and those with curved mouth parts. On the left, we have Proctacanthus philadelphicus with that straight margin, and there's about 18 species represented in the continental US. And then on the other side, we have Proctacanthus haini with that curved margin, and there's only about 4 of those represented in the continental US. So for this question, I decided to focus on those with straight mouth parts because they are more heavily represented. And then also, I figured I could split both of these because in preliminary trees and phylogenetic analysis, these two groups are coming out completely separate from one another and are individual groups. Our next step was to gather locality records, and this step took a bit. So we have a lot of records. Here are points for each color represents a different species. We have these records are from over 190 repositories of insect data, whether that be private or larger collections. And then we also had a total of 6,209 locality records, many of which represent multiple individuals. So a total of 10,000 specimens, and that's only counting those with good enough data to be represented. We also did some fieldwork to focus on areas of lower representation and also some taxa that we needed DNA for later analysis. Our next step was to gather ecological and climate layers, and for this we used WorldClim's BioClim dataset as well as WorldClim's elevation dataset. Finally, we did some niche modeling, and this is the fun part. For this, we used MaxN, or maximum entropy, which we used because it is focused on presence-only data, which museum data is, uniquely only presence data. And then also because of its robustness with other packages and whatnot that could work with it, which, speaking of which, we also used ENM eval to account for spatial autocorrelation by geographic region to help account for some collection bias, then also SDM-tune to further reduce collection bias, as well as to do some data-driven variable selection, and then also hyperparameter tuning and checking throughout the model space for an optimal model using AUC as a metric. And then once we had our models in hand, we were then able to use ENM eval and ENM tools to calculate some different metrics of niche overlap, as well as identity tests between taxa of interest. Here are our maps. Aren't they pretty? They're a little small. I guess if you're watching this later, you can pause and look at them if you want. But these maps, they're a little, so we will be going into more specifics with them in a minute. So on the left here, we have our AUC scores, or the area under ROC curve. And these scores it's a metric from 0 to 1 that basically represents how much how good the model is at predicting true positives based on how MaxN works. And if you have questions on that, feel free to reach out. But most of these are pretty good, right? They're nearing towards one, except for one, and that would be Proctacanthus milbertii. Milbertii has an AUC of 0.85, not bad, but not as good as any of our others. But knowing what I know about milbertii, I know that milbertii is a generalist. It's known that if you want to collect milbertii, you don't have to be as specific with habitat preferences as you do with the other Proctacanthus. It's just more common. It's kind of medium bodied. And like you can see, these flies are pretty well distributed. But what's really interesting to me are these hot spots here in Utah. And so using this model, you might maybe saying, oh, well, it's predictive. So that doesn't necessarily mean that those are occurrence points. However, if we look into the literature, in the Robber Flies of Utah by Riley Nelson in 1987, we have some points for milbertii. And although these specimens have been lost, we don't know where they are, so I can't validate them. Um, it seems that there is habitable areas of Utah, and so perhaps these records are correct. And so I know where I will be collecting next summer. And so we've kind of answered, it seems to predict the ranges of these flies quite well. And even when it seems to not, that predictive power seems to be working in our favor in regards to like older literature. And then also, so our second question, can we use this modeling to aid in delineating species? And so I'm going to highlight these two. We have potential species C, aptly named, and then Proctacanthus longus. Longus is found primarily in Florida, whereas potential species C has a real, just is really out in the sand dunes of Texas. And these two are morphologically quite similar. And it's hard to tell in the picture due to some color grading, and also specimens can grease. But these two species, or these two potential species, I suppose, are extremely hard to tell apart. But with a key, a preliminary key that I have, I can tease them apart without geographic data. So my question is, are they fulfilling the same ecological niche? And so when we look at our models, this is what we see visually. There's a lot going on here. So both of them have a pretty high AUC score. They both seem to have potential regions in Texas that are good for them, but potential species C does not have as much of a presence in Florida, or a hypothetical presence in Florida. We also have these two values, Hellinger's I and Schoner's D. And both of these values are just metrics of niche overlap. And you can see Hellinger's I is quite high and Schoner's D is more mid. But these values are similar to other ones that share geographic space within my pairwise comparisons of non-contested species. So I decided to take it a step farther. And this is the question I want to answer. So this is what this identity test is helping us answer. Hellinger's I and Schoner's D, you can see here this blue line is the empirical data. So this is the actual value of our pairwise comparison. And then this gray normal curve off to the side is the null distribution for if these two species do not share the same niche. And as you can see, they're quite different from one another. What it does is it randomizes all of the locality information to pretend that they are basically within the same area, and then does the Schoner's D and Hellinger's I calculations again. So we have a pretty decent p-value. And so because of this, we can reject the null hypothesis that Proctacanthus longus and potential new species C share the same ecological niche, which is quite exciting. So why is this so important? Well, number one, we just talked about it. These models help highlight lineages and populations that should be investigated further. If these two are physically similar, but in very different spaces and fulfilling different niches, that is more than enough evidence for me to want to continue to research them. Also, I think it's always important to remember museum specimens offer new and insightful data, especially when used correctly. And then knowing an organism's biology can assist in model interpretation. I would argue no model should be interpreted without organism's biology in mind. If I didn't know about milbertii, then I might just go, oh, well, it just didn't predict it very well. But knowing milbertii's generalist behaviors and how AUC reacts to those behaviors was extraordinarily helpful. So in the future, I'd like to include the Kirk mouthpart group. That's about 4 species. You just have to get the locality records for them. I would like to further compare niche requirements between species to identify different strategies within the genus. You can see Coccollettii over here, extremely specialized, versus Milbertii, our favorite generalist. And then also I would like to use genetic data, which we already have, to support or reject potential species C. And so that will be coming soon, so keep an eye on. And then finally, I would like to integrate iNaturalist. Like I said, these flies are huge. They are often photographed. There's a large amount of people online who are talking about these flies, and they have websites about these flies. And so I would like to see how integrating this data, integrating this like public source data will change or will not change these models to see if the source of data that we're using has an effect on our max-end models. So I want to acknowledge some super awesome people and then also some super awesome organizations who have really helped me along the way, whether it be for collecting or for taking pictures. And here are my citations. Any questions? You can message me or email me. So, bye.